Regulation of ionic calcium in cytosol is a very important topic. In this series, we will learn about the slight increase or decrease in the calcium ion in extracellular fluid can cause extreme immediate physiological effect, and how it does or how it regulated is a concern of this. Now the calcium homeostasis. If you want to learn this homeostasis, remember the formula of 3. Now the extracellular ion concentration determined by calcium absorption from the intestine, renal excretion of the calcium, and bone uptake and release of the calcium. And it is regulated by 3 hormones: vitamin D, parathyroid hormone, and calcitonin. Key role is very important in all three types of the muscles, that is skeletal, cardiac, and smooth muscles. It is also responsible, besides the muscle, in the clotting, blood clotting, and transmission of nerve impulses. So the neurons are very sensitive to changes in the calcium in concentration. Whenever there is a hypercalcemia, you will find progressive depression. And whenever there is a hypocalcemia, means there is a decrease in calcium ion, then you will find it become more excited. So what is a very important feature of extracellular calcium? That only 0.1% of the total body calcium is present in extracellular fluid, and about 1% inside the cell, and the rest 99% it is stored in bone. So calcium in plasma percentage wise is only 0.1%, and it is very important. Now out of this 1%, 0.1%, 40% of the calcium combined form, and it is non-diffusible. 10% diffusible and non-ionic form, un-ionic form. The remaining 50% of the calcium in the plasma is both diffusible and ionic. So by the pictogram, you can see that the calcium, this is the most important, is the ionized calcium, that is 50%. Calcium which is combined with an ion is diffusible, and it is non-diffusible. 40% that is combined with albumin is not. Next slide. This is a very important slide for the regulation of ionic calcium in cytosol. Now first we understand why it is very important. You must understand, first point is the concentration gradient between the extracellular fluid and intracellular fluid is 12,000 times. It means its concentration of ionic calcium, it is 100 nanomoles per liter intracellularly and 12 lakhs nanomole per liter outside the cell, means in extracellular fluid. So calcium concentration, or you can say the movement of the calcium, is invert, or gradient is invert, or electrical current moves invert very easily. So there are the 3 channels: voltage gated channel number 1, second is ligand gated, third is SOCCS, store operated calcium channels. Now these 3 channels by which it can enter, calcium can enter inside the cell, or it can release from the storehouse. So during the release, or during the entry of the calcium, or during the activation of the calcium, here you will find IP3 act as a second messenger which is present in the membrane, which activate as ligand-gated channels, and calcium is released from the main storehouse, that is endoplasmic reticulum. So it, after the release of the IP3 second messenger, it activates calcium release from endoplasmic reticulum, and the calcium in the cytosol act as a second messenger. Second point we must remember, besides the concentration gradient outside and inside, there is a gradient also present, or you can say concentration is different from the site of the store or release and at the site away from the release of the calcium or entry of the calcium, you can say. So by the diffusion, when by the diffuses, the calcium may diffuse outside or it may diffuse inside the sarcoplasmic reticulum, or there is an exchanger, sodium calcium exchanger. So by this you can find the gradient is developed. It is here less developed and here it is more concentrated. So there are the three storehouses, endoplasmic reticulum, mitochondria and calcium binding proteins, by which the calcium can be released whenever there is a requirement of the calcium. And whenever there is a calcium inside the cytosol and it is more in concentration, it can release or moves out through the plasma membrane ATPase. Second is exchanger. Third is sarcoplasmic reticulum. So how it goes inside the sarcoplasmic reticulum? By the calcium ATPase, that is known as SARCA pump, that is the sarcoplasmic endoplasmic reticulum calcium pump. So by this diagram it is very clear that if we remember the formula of 3, here is 3 channels, there are the 3 storehouse, there are the 3 ways by which the calcium can be moved out, and we can find out the regulation of ionic calcium inside the cytosol. Thank you.